Right. Assalamualaikum. Good morning, everyone.
Morning, Doctor. Morning, Doctor.
Okay. So let's review. Okay. So previously we have talked about in the first module we have talked about the importance of communication, right? And then in the second module we have talked about what was the second module? Yeah. Second module, we talk about understanding fundamental research concepts. Okay. So why we talk about research? Because we are talking about science. We are scientists. So we have to know what are the importance of, what's the fundamental concepts in research in order to communicate effectively. Okay. Right. In the 10th module, or during our third session, it's about poster preparation and presentation. Why poster? Poster is one of the ways of communicating research or communicating science, right? So I suppose you have gone through the instruction and you have also read the paper and start doing your assignment, okay? So in today's session, we are going to cover the next modules. I hope that I can cover 3 and 4. The third one is not that we we have covered some of the important points in module 2. So module 3 is more about some revision, okay? And I think the main part for today is module 4, which is thesis research project. Okay, module 3 is about modern scientific investigation, and the fourth module is thesis research project. So I have uploaded the modules, the lecture notes last night. Yeah. So I hope you will check every time before the class the lecture notes if you want to have the lecture notes with you during the lecture. Okay, so let's get started. Module 3. Okay, module 3. The topic is steps in a modern scientific investigation. I think we have covered some of the points, so I'll just go through the flow, and then I would emphasize on certain important concepts. Yeah? All right, so when we talk about modern scientific investigation, why it says modern there, why the word modern is highlighted is because when we talk about science, science does not exist only in 2022. Science does not exist in 2000. It has existed for more than years, for more than 100 years even, right? Like 100 years ago. But in the past, our ancestors, they practice science, but maybe they do not know that that is actually science, or even biotech, right? Biotechnology, like people have produced great beer, kimchi, whatsoever, the fermented food. That is biotechnology, but they may not know that the field is coined as biotech after many years they have created that. Okay, so what's the difference between science today and science in the past? There are certain things that differentiated it, and probably because of nowadays we are more knowledgeable because of the information is vast, right? There's a lot, and then we have mastered certain skills, technologies, right? So meaning to say that the science that we practice today might be in a better way, in a more organized and systematic way, rather than those science that have been practiced many, many years ago. Okay, maybe there are some things that need to be improved in the past. Okay, so when we talk about modern scientific investigation, the scientists, they have outlined several steps that need to be taken before we start any scientific investigation. Okay, so it is because we want to make sure that everything must be aligned with the scientific rules. Okay, for example, we must know the problem first. We must design the experiment. We must carry out the experiment, and then after the experiment, we must analyze the data before we can verify or process the evidence. So there are certain steps. So you cannot just jump, like you have an idea today and you want to jump directly to the lab tomorrow. That could give you lots of hassles, because there are many things that need to be planned before you enter the lab. For example, when you want to start your final year project, you cannot get the title and go directly to the lab. You have to read, you have to understand the topic, you have to know what's the gap. What's the gap? You know what still remember? Research gap between your proposed research and the research that have been done in the past. Okay, and then you have to know what you want to focus on. You cannot do everything because of the time frame, limited time frame. So you have to focus on several things. You have to outline several objectives, right? So that's the thing that you have to plan. And also, we train you in the FYP to write a proposal. So that is actually part of the steps in a modern scientific investigation, where you have to, after like doing the literature review, you have to come up with a research proposal. So that research proposal shows that you have done several systematic steps whereby you do some research in reading, you do some research in finding the methods, and so on. Okay. Now, so let's have a look at the steps in a modern scientific investigation. First, of course, you have to get the topic, or know the problem that you want to solve. Okay, identify the question, define the research problem. We're talking about research here. And then after you know what you want to focus on, so you have to form hypothesis, right? Hypothesis. So we know already that I've learned in statistics, there are two hypotheses: null hypothesis, alternative hypothesis, right? It means that you must have some ideas or some expectation what you will get before you start the research. Okay, this is still before the research is done. Yeah, and then from the hypothesis, you can predict possible observations or results. So it can be like this, it can be like that, it can be this and that. So you are ready to face it. Right, so after that, you have to design an experiment. So I've talked about designing an experiment. So what does designing an experiment means? It means that you have to know what are the variables, parameters that you want to study, right? Variables are the things that you control, or it could be like you fix it, or you can vary it. So that's why it's called variable. The vary means that, let's say, temperature. Temperature. Symbol for temperature is capital T. Okay? So the temperature, if you want to vary it, means that you want to control it. You want to control at a certain level. Okay. So you might vary it at maybe 30 degrees, 40, 50, or 60. Okay. These are the levels of the parameters or variables that you want to test, for example. This is an example. And then after you have identified the variables, you have to determine the procedure of your experiment. Procedure means the methods. Methodology is the methods that you want to use. Because in doing certain experiment, there are certain methods. So how you want to know the methods? Of course, you are a novice, right? You do not invent yet any method, but you can refer to the methods that have been reported by other researchers in their articles, research articles, or research journal papers. Okay, so you have to decide Okay, maybe this experiment you use this method. Why you use that method, you have to know why. Okay, that one is your task to know why. But the methods you have to refer to those established in the literature. Okay, so after you design an experiment, of course, for the FYP, you have to write a proposal, and only then you can carry out the experiment, right? Carry out the experiment means you enter the lab with the plan that you have. Okay, of course, you have jotted it down in your logbook, the plan, the methods, and so on. And once you carry out the experiment is going on, and you get some data from the experiment. So it means that it's time for you to collect the data. And just collect the data, you also have to analyze the data. So collecting and analyzing data are two different things. Okay. So please understand this, collect and analyze. Collect means you just take, let's say your experiment is dealing with protein quantification. You need to quantify the amount of protein. So how you do that? You use Bradford method, for example. And then in that method, you have to do some reaction between your sample and also the reagent. For example, we use Bradford reagent. And then the reaction mixture, we have to measure the absorbance. Absorbance is an example of indicator that can show the difference in terms of the reaction mixture, the color of the reaction mixture. So when you measure the absorbance, you have to use a spectrophotometer, right? So that's the name of the equipment. So it means that you collect the data, you measure the absorbance, you get the data. That is the process of collecting the data. You get the data from the lab, but you're not yet into analyzing. Collecting means you do not know whether your results is okay or not. Maybe based on certain experience, if you see the change of the color, it becomes more intense, then you know that maybe absorbance might be increasing, so the protein might be more. So that is based on physical observation. But you have to analyze your data, means that you have to maybe calculate certain things based on the formula, right? And also you have to analyze the data in terms of the statistics, like what is the mean. Let's say you have triplicates, three different things that you measure, but of course you need to get the average reading for all the three, right? So you have to calculate the mean, you have to find the standard deviation. So that is statistical analysis. Okay, so that is analysis. So once you analyze your data and you see the trend is like this, and you plot, analyze the data analysis is also, it also involves data plotting. Means that once it depends on the types of data. If let's say your data is quantitative data, also means that you have the numbers, the values, and you can plot it. Okay? Maybe for absorbance, you have to plot it. So part of the data analysis is the data plotting, okay? Or you create a graph, or maybe you create a table to show how it varies, for example. So that kind of things are included in data analysis. Okay, and normally you do the data analysis once you have the results, and you do it maybe at home, or maybe like not necessarily in the lab. Okay, sometimes it might be in the lab, sometimes you can do it outside the lab, because you are dealing with computer and statistical software. Okay, once you get the data, once you have analyzed the data, and that is when you process the evidence. So when you process the evidence, means that you want to see whether your results support or disapprove your hypothesis. What does it mean? Okay. Do you achieve your objective? Okay. If let's say you're looking at the best temperature, for example, from 30 to 60, do you get the difference? Do you get the best temperature? Which one is the best? Things like that. Okay, so that is the evidence interpretation. Okay, and that is when you make a conclusion. Conclusion about your work at that point. Okay, whether you achieve or not your aim. So that are the learned steps in a modern scientific investigation. Okay, I just go through very briefly, yeah, because I think I have repeated some of the points in my last lecture, like research problem. You have to know what's the research problem. So I just want to emphasize few terminologies that you need to understand. Research problem, that's one of it. And then there is also research gap, okay, research gap, problem statement. So these terms are very popular in research, or in FYP, you need to know what it means. Otherwise, if let's say the examiner asks you, what's the research gap of your project? So before you understand your project, you have to know first what the research gap means. Yes? Problem statements, and then yeah. So research problem is like, of course, the problem of the research, like the research that you are focusing on. What are the things that you want to solve? Okay, of course, there is a problem. Problem means the things that have not yet been discussed, the things that have not yet been studied before. Okay? That's why you are interested to do that. So that is the first step in any research. Okay. So research gap is the difference between your research and other research. Remember when I introduced you the diagram last time? So let's say this is your research, and this is other research, okay, maybe A, B, C, and D. So you can see that there are overlap, right? Overlap regions means that the things that are similar to what you want to focus on, but at the same time, there are areas that are not overlap with any research, which is the area in the middle here, right? So in the middle here, which are the focus of your research. So this is actually the research gap, not the overlap one, because there is a gap for difference. Yeah? Okay, so that is research gap. Problem statements. Problem statements are statements, sentences, or statements that indicate the problem. Okay, so it's like associated with this actually, but it's just a term that you also have to know. Okay, so that's a research problem. Device or form hypothesis, I've talked about this as well. So you have to know there are two types of hypothesis, null and alternative hypothesis. Null, alternative. Okay, so what's the difference between those two? Null means there is nothing happen. You do the research, but there's no change. It could be one of the outcomes. But alternative hypothesis is the other way around. You do the research, there is something happen, which is actually the motivation of your research. If you just believe that it's only null hypothesis, so what's the point of doing the research, isn't it? So most of the time, the motivation is more towards is more because of the alternative hypothesis, because you believe that there is a change and it is worth to do the research. Okay, right. You revise by yourself, yeah, null and alternative. I think you have talked about this. You have discussed about this in statistics last semester. Okay, from the hypothesis, predict possible observations and results. Okay. Design an experiment that tests the hypothesis by trying to falsify your prediction. Identifying, controlling your variables, determining the procedure of your experiment. So I've talked about designing an experiment. In the last lecture, it's about planning. Yeah, it's about planning actually. Planning your work. Okay. So when you say you plan, you involve a lot of things. You have to know the methods, you have to know the equipments that you're going to use. Maybe like if let's say you want to decide which methods, you have to read more in order to differentiate the methods. Okay. And not just that, you also have to know the risk of the work. You know the risk of the works. For example, if let's say you are using certain chemicals, especially chemicals and equipments, you have to know, let's say your analysis involve IPTG. IPTG is an example of chemical that can induce the protein expression. So before you want to use any chemicals in the lab, you have to analyze the risk of the chemicals, how you want to handle the chemicals, because some of the a lot of chemicals actually are hazardous if you don't handle it properly. Okay, for example, IPTG is an cancerous agent, so you cannot touch it with your hands. So without knowing the risk of that, if let's say you do not know the background of the chemicals, the chemical can cause cancer, but you do not know about it because you don't do the risk assessment. So this is another thing that you have to know, risk assessment. So what happened? It will endanger you, not just you, but also people around you. Okay, that is in terms of chemicals, and there are a lot, not just IPTG. Chenomycin. Okay? Some of them may affect the infertility. So be careful with that. Some of the chemicals are explosive. So explosive, you have to handle it under the not laminar flow, is under the fume hood. Okay, so you have to identify the equipments in the lab. What's fume hood is used for? Fume hood is a hood where you use whenever you want to handle any chemicals that are that can cause you, like it's hazardous for you to inhale, especially base, and also all those chemicals that can suck you up when you inhale it. Okay, so that's why you use fume hood. And then equipments, for example, equipments. Before you use any equipments like autoclave. Have you heard about autoclave before? Autoclave? Have you heard autoclave? Have you used autoclave before? Hey guys, are you with me? Have you heard autoclave?
Yes.
Yes. Okay, so what's autoclave is used for? Ngantuk ke pagi-pagi ni?
Sterilize the equipment.
Yes, correct. So autoclave is used for sterilizing equipment. But before you use autoclave, of course you have to use autoclave, but before you use it, you have to know how to use it and what are the do's and don'ts. Okay, one of the don'ts is if let's say you want to autoclave bottles, right? Bottles, especially with whether it's empty or whether it's with liquid inside, you cannot tie it. You cannot seal it tightly. Means that you have to lose a little bit the caps, right? So why? Is because autoclave it involves high pressure. So whenever the things it's heated inside and then it's pressurized. So anything that like with anything inside closed container, it will have high pressure, isn't it? So if let's say you tie it hard, then what happens is that it will break. The bottle will break, and the chemicals will come out. Okay, so that's why you have to lose it, and don't fill it too much, too full. For example, this is a Scott bottle. Okay, so you have to these caps, you have to lose it. You have to close it still, but then you have to lose it. Don't tie it, and then this one you cannot fill until the neck of the bottle. So at least it's like 80% of the total volume. So things like that you have to know. How to know? You have to read. You have to identify the equipments that you want to use, and you have to also assess the risk. Okay, so when the things break in the autoclave, so what happened? So there are so many problems. If let's say the equipments are broken, okay, so it will delay your work and other works. And also not just that, actually, it will actually give you hazards in the lab. So those things that we do not want. So part of the experimental design is also assessing the risk. Yeah? And it's very important because some of the well-established universities, they will not allow you to enter the lab without you filling up the risk assessment form. So that is a way how they can ensure that you have assessed the risk associated with your work. All right. Good. So that's part of it. Now, so once you have designed the experiment, okay, and you know what you want to do, you know what the methods that you want to use, procedure and everything. So when you start your work, you carry out your research. So you have to know few things more, right? So you have to know what kind of research you are doing. Okay, so there are various types of experimental research. Okay, there are controlled scientific experiment, correlational studies, and also part of the experimental research is data collection and analysis. And then there is also computer modeling. So basically, data collection and analysis is compulsory for all, but the rest of these, the other three, it depends on your work. It depends on your study. So some studies, they are categorized under the first one, or some studies are correlational studies, or some studies, they involve computer. So they involve certain software and use computer. Okay, so what is meant by controlled scientific experiment? When you have variables and you vary it, and you have something else that also you control, and maybe you keep fixed or you fix it. Okay, so when we talk about variables, there are different types of variables: independent variable, dependent variable, and control variable. So independent variable are those variables that you change. Yep, you change. And for example, whatever the things that you want to study, like temperature, pH, you change it because you want to see the effects. And the effects are, for example, the protein concentration, the cell concentration. So those things are what you observe. So those variable, like let's say protein concentration, cell concentration, so they are dependent variable. So they depend on the independent variable. They depend on the independent variable, such as temperature, pH. So at low temperature, there might be maybe low protein. I don't know, depends on the microbes, or low concentration of cell. So it depends. That's why it are dependent. Control variables means those variable that you fix at the same, you fix the same throughout the experiment. Maybe it's not your focus. For example, you just want to study the effect of temperature and pH. So you fix maybe the glucose or the substrate concentration. Okay, means you fix for all experiments, or maybe you fix throughout the experiment. Okay, so that's control variable. Correlational studies, studies that are related with two things. Maybe you want to see what's the correlation or relationship between TSP1 gene expression and tumor growth metastasis. So it means that you have two variables, and when you do the experiment, you want to see how it correlates to each other. And at the end, it will be presented in graphs like this. It could be, the results could be positive correlation, means that, let's say this is variable 1 and this is variable 2. So it's increasing proportionally. Yeah? That is positive. Negative correlation means if, let's say, someone is going up, another one is going down. For example, this one, when it decreasing, this one increasing, right? So this is negative correlation. But if you don't have any trend, any specific trend of the relationship, yeah, something like this, which is arbitrary or abstract, you can say. So it means that there is no correlation. Okay, so these are the possible conclusions from this kind of study. The data collection, I've talked about it just now. You collect the data, you analyze the data. So it's compulsory in any type of research. Computer modeling, you use mathematical or certain software to see the simulation. Simulation means that you maybe have like certain data, but you want to see maybe if let's say you extend certain things, what happened, right? So it's more to like prediction of the existing process. Okay, so but you need to use certain software to use it. So these are examples of results from a software for modeling a biological process. Okay, it's just an introduction. You don't have to understand why the color is this. It's normally sometimes it gives there are certain meanings. Like for example, this red is the highest, maybe like here, yeah, is the highest region. So followed by blue is the lowest region. So that's the interpretation of it. Okay? So in order to use, or in order for you to start in this kind of research, you have to know how to use the software. There are certain softwares for certain applications, certain studies, and you also have to know how to interpret, not just use, but how to interpret. Interpret means like if you have this kind of results, this kind of red, yellow, and this kind of graphs, 3D graphs. What does it mean? Okay, so you have to understand how to interpret as well. Okay, select the data. Yeah, I think I've introduced you. There are two types of data, qualitative and quantitative. Quantitative relates to number. Qualitative means features, okay, traits, characteristics, whether the shape of the bacteria, maybe the morphology of the plant, what you can see, how many leaves, for example, the physical characteristics. Quantitative is something related to number, and normally you can graph it, yeah, and you can put like plot a graph or a bar chart or pie chart because you have the number. Okay, for example, this one is qualitative data. This is STS phage gel. Is a result that shows protein separation. So it means that this one is one protein, this is another protein. So of course you have to also know how to interpret this. But this is an example of qualitative data. Okay. Okay, when we talk about statistics, they are descriptive statistics and inferential statistics. Descriptive statistics when you describe whatever the things, whatever the existing traits. For example, you get the results and this is, you describe it. Like maybe you say what's the mean, what's the deviation, what's the variance. But when we talk about inferential statistics is related to prediction. Okay, prediction or forecast. Prediction. Forecast. So it means that you have the results and you use that results. Oh, sorry, no. You have the data and you use the data to predict maybe how the behavior of the cells. Maybe if let's say you have this kind of condition, okay, or maybe prediction of the reaction of certain enzymes or whatever. Okay, so it's something on prediction or forecast. Okay, so that's inferential statistics. So if you are dealing with just now when I mentioned computer modeling, that is an example of inferential statistics. Yeah, so you are predicting or forecasting. And the final step in a modern scientific investigation is to process the evidence. So this is when you have the results already. You have analyzed the data and you get how the data, the trend looks like, and so on. So that is when you process the evidence. So when you process the evidence means you want to check whether your results support or not your hypothesis, whether you achieve the objectives that you have outlined or not, right? So what is the conclusion? Yeah. So that is where you process the evidence and you must come up with conclusions. And also recommendation, because in research there is no endpoint, meaning to say that once you do your FYP later, the nature, of course, the FYP, yeah, of course, you finish the FYP by semester 2, that's the end of your FYP. But when we talk about the research itself, right, the study itself, there is no endpoint for it. Maybe someone will continue that study, the next FYP batch, or other students or other researchers, maybe if when they read your report, your article, they have some ideas, some more ideas, new ideas. So they will continue from that point and do something else. So the nature of the research is never ending. Okay, so that is why after you have finished certain work, certain scope of work, you must have some recommendation, because you have gone through the process, and of course you cannot do everything. So if, let's say, if you want to recommend to others, okay, you should do this, you should continue this. So there must be some recommendation from you as the person who carry out the research. Okay, so at the end of the process, at the experimental, at the end of the scientific investigation, of course you must have the conclusion, and you must also have some reflections or some ideas on the recommendations. Okay, so that's why in the last chapter of your thesis, final thesis, FYP2, you must include these two components. The first one is conclusion, and the second one is recommendations. And recommendation is actually where the examiner will see how far you have understood the project, how far you have actually developed your research skill. Research skill here means that you have reflected, you have, you become more mature after you have done the research. You know something, but you want to suggest something. Okay? That is actually very important. Okay, and the output is, of course, in terms of the thesis. So once you finish the FYP, controlling FYP2, you will produce your thesis. Maybe if, let's say, your results are good, you can publish an article, you can present in a conference, you can maybe present in terms of poster. Okay, so there are many outputs that can arise from your work, from any scientific investigation. Okay, so that's module 3. Okay, so the steps in a modern scientific investigation. Okay, any question for this before we move to module 4? I have to save, okay, so far good, okay, thank you. I have to save my voice because I have another lecture this afternoon. I cannot drink today. So I hope that I can finish module 4 briefly too. Okay, maybe I just emphasize on certain important points. Ni dah penat juga ni, setengah jam cakap. Okay, module 4. Module 4 is about thesis research project. Okay, why it is named so? Because when we talk about research project alone, research project, it can be of many types. Okay, we understand thesis. Thesis is a document that that is sent because of a research has been done. Okay, a research has been done. For example, FYP, MSc research. MSc is a Master of Science, and then PhD research. Okay, so all of this type of research produce thesis, produce these documents, because the research needs to be examined. Like, we need to examine your FYP, so that's why you have to submit your thesis. For master student, after 2 years of study or 3 years, they have to submit a thesis, because we want to know whether their MSc research is correct or not. Same goes to PhD research. So all of this produce thesis, right? And this type of research is called thesis research project. This type of research project is called thesis research project. So they are research projects that are not thesis based. For example, like now, I already done my FYP. I have already done my MSc. I've already done my PhD. So if, let's say, I want to carry out a research project, okay, I don't call that research project as a thesis research project. Of course, if from the student's point of view, if, let's say, I have students, I supervise a student, from that student's point of view, of course, his or her research is thesis research project. But from my point of view, the project is not thesis based, okay, because I don't have to submit a thesis. I myself do not have to submit a thesis. So for your side, because you have to do FYP later, so you have to know your project is one example of thesis research project. Okay, so that's what it means. So here we would see some of the topics. Okay, why students undertake research study and write thesis. What are the steps in carrying out research? Principle of research, characteristics of excellent researchers, and behaviors and ethics in research. So I think why must students undertake research is I talked about it just now. So because you need to fulfill the requirement of a degree. Bachelor's degree, FYP is part of the degree. It carries 7% on, even though it's only 7%, but you have to pass it. You have to fulfill it. FYP 1 is 3 credit hour, and FYP 2 is 5 credit hour. 5%, it's not 5%. I think, yeah, 5. So it means that, but I think it will affect a lot your GPA or CGPA, especially FYP 2, 5 credit hour. So you have to focus on it, try to get A or A minus, because it will affect a lot your CGPA. So that's bachelor degree. Master degree. So master is divided into coursework or research. Okay, for research based, they have to submit a thesis at the end of their research period. Yeah? So that thesis will be examined by examiners, and the student also have to present and defend their thesis in a viva voce, the name of the exam, oral exam. For the FYP is the oral presentation. Yeah. So it's for viva, but viva is more commonly used for postgraduate. And then for the PhD as well, it's the same. Okay? The difference between master and PhD is the period of studies and also the depth of the studies. Master only takes 2 years, so it means that the scope is smaller, whereas PhD takes around 4 years. Means the amount of work and the amount of knowledge to be sought for is more for PhD. Okay, so all of these produce thesis. So that's why what thesis means, you have to go through all this process and write thesis at the end. Okay, so what are the advantages once you undertake research study and write thesis? So means that once you start a research, throughout that research period, because that research period is not the same as any course that you have taken before. Like, for example, you take courses, that one is called coursework. Means we provide you the outline like this. We give you even the lecture notes, and we have exams at the end. So it's structured. Yeah, so it's coursework. But research is something that you have to you have to rely on your own, like means that you start the research in the lab, whatever the things that you get in the lab, that will actually develop at the end your thesis. Means like the whole process is more independent. But what it means, it will actually develop a lot of skills in the person, in the researchers, or in the students. Okay, so the process will train you to think more, because in the lab, you cannot rely on the supervisor all the time. You have to think by yourself as well. Okay, and you have to apply your common sense sometimes. Thinking ability is one of it. You have to be systematic method of thinking. It means that as scientists, you are actually trained to be systematic in your work, in your planning. You cannot jump to do the experiments straight away, right? So you have to do the steps. So that's also a skill, okay? And believe it or not, once you practice all these things, all these skills, in other things outside the lab or in your life, you will also be trained to be systematic. Like the way how you think will be different, I can say. And especially after some time, let's say you have done your FYP 6 months, and during that 6 months, you are trained to be systematic. Like you cannot do things blindly, and you have to follow the steps. And what's more, if let's say you do master for 2 years, you are trained to be in that sort of in that sort of environment. And let's say you plus another PhD, 4 years. And at the end, after like maybe 7 years, yeah, so the way how you behave or work will be different. Okay, so that are the advantages of doing research. So this is the points here. Systematic method of thinking, improve understanding of the scientific methods. Of course, in the lab, you learn a lot and you see a lot, you observe a lot. So it's not just about what you read from the papers, it's what you understand. Your understanding is also enhanced by what you see, what you observe, what you observe, right? So when you see and when you do the things by yourself, you will understand better. Yeah? So that's improving the understanding. Harness logical reasoning ability. Okay, so it's something to do with like when you say, for example, the change of the colors or maybe the viscosity of the culture is maybe increasing. Viscosity means kelikatan, like it becomes more viscous. Yeah. So what it means? So it means that maybe the cells are more, something like that. So that is logical reasoning. You have to think about it. And you also have to be always be curious in the lab, because that will lead you to think and find the reason. Okay, means if you see something change, you cannot just observe it and let it be like that. So you have to think about why the change is like this. What does it mean? And you have to relate with what you have studied. Yeah? So that is where you apply all this logical reasoning skill. Develop expertise in the field of choice. Okay, so biotechnology is a huge field. Even though it's a branch of science, it's already like at this point you can see that it's already a focus field. But actually under biotech there are a lot of branches. Animal biotechnology, plant biotechnology, industrial biotech, microbiology, molecular biology. So when you do your FYP, you are focusing in one of the areas. Okay, means that if let's say you take a project under animal biotechnology, means you are going to focus on the work in animal biotech. So at the end of your FYP, you're going to know more about animal biotech than other niche areas. Okay, what's the importance of it? You will develop your knowledge in that field. Means that you create something, you develop your expertise. Okay, so that is actually the purpose of research. It's a focused research study where you will develop your expertise in that field. Okay, so you imagine if let's say you further your study after MSc, you focus, let's say, more on animal, you focus on other things or other parts of animal biotech, and then you proceed with PhD, means that the depth of knowledge that you have in that field will be actually more than those who just maybe after the first degree, right? Okay, so that is actually, it means like, it's like this actually, like at this time, you study this, you study this, you study this, you study this, right? So this is biotech. Okay, so this is before FYP, maybe. Before FYP. So means that you study on the surface. You know a little bit about plant, you know a little bit about animal biotech, a little bit about industrial biotech, medical biotech. But it's just like your knowledge is about the same for all. You don't know really much, you just know on the surface. But let's say this is you study a little bit, you take FYP, yeah, FYP. So means that this one is maybe fermentation technology. Fermentation technology. So you study a little deeper than others, but others they have they will study other fields lah. This is like, let's say here, you are here. So after you finish your IP, you know a little bit, right? A little bit, then the rest. So let's say MSc. MSc is 2 years. You pile a little deeper, right? Maybe in the same field. So here you are. After you finish your MSc, you will know, this is just imagine this is like the knowledge, your expertise that you have gained, expertise, the skill in the lab, in fermentation, right? This is MSc. So this is where you are. This is after FYP. So if let's say you proceed again, PhD, right? PhD is another period. So this is PhD. PhD means Doctor of Philosophy. Okay? So your knowledge will be increased more, right? So in terms of the depth of knowledge, rather than here, this is surface. Okay, so that's the difference. And this is what we call expertise, like your expertise, specialization. Like me, I'm focusing on fermentation technology, but I do not know much about animal biotech. I do not know much about plant biotech because I don't choose that niche areas. Okay, so so my advice is before you proceed with your FYP next semester, before the topics are released at the end of this semester, you have to choose the topics, depends on the coordinator. Normally it's at the end of the semester 2, like before you start your next semester. You have to know your interest. You want to proceed with animal biotech, plant biotech, or what? Industrial biotech, medical biotech, microbiology, or what? Okay, so you have to know the branch of biotech by right now. Okay, so that whenever the topics are released later, you don't spend too much time because you have to compete with your friends in terms of the topic, because some of the interesting topics that would be sold out soon than others. Okay, so you have to know first your interest. Okay, so maybe you are more interested with plant biotech. So you have to know who are the lecturers in our program who are specializing in plant biotech. For example, Dr. Ho. So you have to be alert with what Dr. Ho is doing, the research, things like that. So why I advise that, because if let's say you do not know yet your interest, you do not know yet what is the difference between animal biotech, plant biotech, and whenever the topics are released, people are chasing for the topics, you are, you tak dapat lah topik yang you suka. Okay, macam tu. So now you have to be ready in your mind, you have to understand biotech, the branch of biotech, and identify who are teaching those courses. I mean, who are specializing those courses. Like me, I'm in fermentation technology. So the lecturers who are in fermentation technology, myself, Dr. Miki, Dr. Dayang Sarwani, Prof. Awang, okay. So we will release the topics under fermentation technology. So those animal biotech, Dr. Chung, Dr. Lee Kwee Soon, okay. Plant biotech is Dr. Ho, macam tu lah. So you have to know first this one. So once you are ready, okay, maybe you want to focus plant lah, you tak nak lain. Okay, you focus lah dengan topik-topik yang dirilis oleh Dr. Ho macam tu. Okay, alright. So that is expertise in the field of choice. Yeah? Okay, now you know that biotech ada niche areas banyak lah sebenarnya. Actually, what we cover at our program, we don't cover all the niche areas of biotech. There are some other niche areas that we do not have the facilities in UNIMAS because of many factors lah. Tak ada equipment or tak ada lab yang sesuai macam tu. Okay, why must students undertake research and write this? I think I mentioned about it. Okay. Develop expertise, yes. Okay. Develop self-reliance and independence. Okay, this one is ada sikit lah tadi kan. So you'll be independent more in the lab. Okay. What is actually the purpose of supervisor? Supervisor give you topics. I mean, like he or she will pave the path for you. You know, like he will open the way for you. Okay, you enter here, but it doesn't mean that he will actually guide you towards. Well, he will guide you, but not to an extent that he will be next to you all the time. Okay, that's the thing that you have to know when you do your FYP or research. Means that you yourself have to be independent. What it means to be independent, you have to find articles by yourself. For example, you have to do some homeworks by yourself. You have to read this, read that. Okay, once let's say you do not sampai you tak faham betul-betul tak faham, then only then you ask the senior in the lab or your lecturers. Maybe you confuse certain methods macam tu, but you have done some reading. So that kind of things that you have to develop, self-reliance and independence. And then improve writing ability, because writing is part of the research, right? Because once you have done the research and you want to communicate your words, you have to write thesis, you have to write an article. You cannot produce documents that are not of poor quality. So you have to improve your writing ability. And also, having said that, it's actually English, the language, because we're using English. So you have to improve your English as well. Why must students undertake research? Facilitate involvement in networking and teamwork. So once you do the research, you will know more people, right? You will know more researchers. So that's how you create the network, and also how you create the team. Maybe after FYP, you want to do certain research. You are working, and you know certain people in the field. So you want to create a team. So you have to know first before that. It's through research that you know other researchers. Okay. Steps in carrying out research. Okay. This one is actually what we need to do now. Okay, this one is done. I've covered that. Information gathering is literature review. Maknanya you baca artikel, kan? So that process is called literature review. You are reviewing the literature or the articles, and you gather the information at the same time. So then you write the research proposal. This is, in your case for FYP, is FYP proposal. But in any other research project, for example, like myself, okay, I don't do any FYP anymore. Let's say I want to do a new research, I also have to write a research proposal, because it's this step is actually mandatory for all kind of research. But in your case, later you will do your FYP, so your proposal is called FYP proposal. Okay, final year project proposal. So this is when you start doing your work in the lab, FYP2, and at the end of FYP2, you also have to write thesis, FYP thesis. That report is called FYP thesis. Okay. Principles of research. So what are the principles of research? The objective must be meaningful. Means that when you set the objective, it must be something that is that has a meaning. Okay. So we can do research, but sometimes the research might just be something that is to break the curiosity, or maybe less meaningful. So meaningful objective means that it will give benefits to the field. Okay, so something like that. And also it must be reasonable. It must be reasonable within your scope, within your time frame. Let's say you are doing your research in FRST labs. You must make sure that the lab has the equipments that help you to achieve your objective. So if let's say there is no such support, it's not it's not meaningful for you to set that objective. So you have to consider lots of things before you set your objective. Reasonable cost. So the research must be within reasonable cost. So any research actually is funded because the cost of research is not cheap. So we cannot use our own money. We cannot use other fund in university. Not to say we have to use certain specific fund provided to carry out the research. Okay, so that fund is called, this is actually grant, but it actually means money, fund, money. So that money or fund is provided by certain bodies. For example, it could be the university, it could be the government, for example, Ministry of Education, Ministry of Science. So they will provide certain grants for the researchers to carry out the research, or maybe some industry and also overseas grant providers, certain bodies lah. Okay, so when we want to do the research, we have to estimate the cost of the research, especially the leader. For you, FYP, you don't have to think about it, but you have to know that the nature of any research, we have to think about the cost of the research, the overall cost. Let's say the whole project for the 2 years, maybe the amount needed is 200,000 to cover the cost of the chemicals or certain equipments that need to be bought, or the salary of the researcher. Okay, so when you do a master, some of the research projects, they are under grant. So meaning to say that they allocate certain money for the payment of the salary of the MSc student. Okay? Maybe they pay you like 2,000 per month or 1,800 per month. So a part of the cost here, it covers the salary of MSc or PhD student. Okay, so MSc and PhD student, although they sounds like students, but they are actually working, and they are considered as, MSc and PhD are considered as career. Okay, so that's the difference between bachelor's degree and postgraduate study. Okay. Right, that's the course. Appropriate research scope. Yeah, we have to determine the scope of the research. Like for example, number of parameters, number of samples, number of treatments. If it involves sampling, right? If let's say you are taking bacterial sample from certain areas, you have to know where you want to collect it, and how reasonable that area is, and things like that. Vertical or horizontal spread. This one refers to the scope of the research. So vertical means vertical. So if let's say you want to investigate maybe one area only, so you investigate lots of things in one area. So that is like maybe the samples, bacterial samples in one area. That is vertical. But if you are talking about many samples from this area, this area, this area, but not maybe like you don't investigate that really much, right? Certain things, only certain things. So you cover like many parts, that is horizontal. Okay, that's only one example. Could be other example. Okay, I'm learning somebody master, PhD, bachelor's. Principle of research, relevant expertise. Okay, of course, you have to go for supervisor yang ada expertise in that field. Okay, so that's relevant expertise. Scientific standard techniques and procedure. Of course, what is meant by standard techniques and procedure, where to get it, how to know whether they are established one. So get it from reliable sources. So what are the reliable sources for research? It's always journal articles, books, okay, but normally it's articles. Okay, why we refer to this, not just the website, normal website, because these are reviewed. They have been reviewed. They have been verified by experts in those fields. So it means that they are reliable. Okay. So whatever the information from the journals depends on the journals as well. Like some of the journals, they are scam journal, you know. They are also fake journal or journals that are actually they just think about profit. But those journals need to be screened as well. But those good journals, they produce good articles and reliable information. So that's where you get the standard techniques and procedure, the methods. Okay, responsibility. Parts of the principle of research, of course, the responsibility of the researcher. Okay, so for example, if I am the principal researcher of the project, I have to be responsible with the execution of the project. And the members of the project as well, they have to be responsible by contributing, for example. The students, let's say the student is the one part of the members as well. The students have to also do their parts, do the research. Responsibility, characteristics of excellent researchers. Okay, these are the four. I think you will practice this whenever you enter the lab next semester. Okay, so this is just your theoretical information. Yeah, so what an excellent researcher should be. You must skill means that you have to develop your skills. You must be knowledgeable. How to be knowledgeable? You have to read a lot. Okay. Intelligent, when you are skilled, when you are knowledgeable, first you will develop the intelligence. Okay, that's what I can say. Motivation, attitude, hardworking. So you have to be disciplined. This is what I think the key thing. It doesn't actually necessarily need you to be, how to say that, you are not going to be automatically intelligent, you know, when you start your research. Like me, when I started my FYP, for example, I do not know anything. But what actually develop you is the attitude. The attitude, discipline, hardworking, diligent, persevering. Those things are easy to be said, but when you experience it, you have to really walk the talk. Okay, so hardworking, what it means. Because FYP, for example, you have to find your own time to be in the lab. You cannot just rely on your supervisor say that, okay, you can enter the lab today. So you don't have to rely on your supervisor. You have to be on your own. You have to be hardworking. You have to find time, available time for you to go to the lab. Okay, you have to be disciplined. If let's say you say that this semester, this week, I have to finish this work. So you have to be disciplined. So those kind of things like diligent, persevering. Persevering means like you have to be resilient in the lab. Why? Because in the lab, there are so many failures. Okay, failures means like your experiment is not working for the first time, second time, third time, and only after fourth time, repeating it, then only then it works. And it is normal for everyone. It's not just you. Okay, so it means that don't be upset when you get the first failure. It's normal. Second failure is normal. Third failure, even fifth, seventh is normal. But what is important? Your attitude. How you want to improve that? How you want to how you want to improve the experiment? What is the mistakes you have to figure out? You have to troubleshoot your work. Okay, so that's why I said the attitude is actually the key to everything. It will develop your skill. It will develop your knowledge. It will develop your intelligence. Even like the first point just now. Okay, so it's all about your attitude. Yeah? So please bear in mind that FYP or research is another field. It's a different field from the coursework. So you have to be ready. You have to have that mindset. You have to be ready, and you have to be disciplined. I have nothing much to say like that. That is all about attitude and discipline. Okay. Forward-looking, proactive, visionary. Proactive means that you have to, when you plan, when you know what you're doing, you must be proactive now. Okay, let's say next week is a break. So you kena make sure lah everything settle before, contohnya before raya ke. You kena settle lah, you kena work proactive lah, you kena tengok plan masa depan tu. Forward looking. So if you know like let's say your supervisor will be on leave for a week. So dalam masa 1 week tu you tak dapat reach your supervisor. So you have to do extra work before that so that you can get some advice before he or she left. So that kind of things are forward looking. So let's say the lab closure, maybe minggu depan mungkin equipment ni tak boleh guna. So you have to plan your work. So you have to that's what I mean by forward looking in the lab. Proactive, visionary. Flexibility, planning skill. Yeah, planning is very important. Time management skill also very important because everything needs plan in the lab. So you have to plan everything, make full use of your logbook. Okay. Before you start any research, you have to have a logbook. What is logbook? Logbook normally is a long book. I think you are familiar with this. You have practiced this kind of logbook keeping, isn't it, in your high school? So whenever you start an experiment, you have to write experiment, the objective, the methods. So you have to practice all these things again during FYP. So why it's associated with planning? Because before you start any work, you have to plan, you have to write down the methods, you have to create maybe certain tables for you to fill out during your data analysis, and you have that you have to prepare up. And it's also associated with the time management because you will know you need to do this, you need to do that, and that will help you to plan in the lab, because in the lab the equipments will be shared. The equipments sometimes you have to book certain equipments. So because there are other users. So means that if let's say you want to do fermentation next week, you have to book the equipment, the shaker, this week, a week before. So that's why time management and planning is very important. So if you don't have planning, if you don't manage your time properly, and suddenly you want to start the experiment, everything's there, and then the equipment is not available, so it will ruin everything. Okay? Because preparation in the lab for the experiment is a lot. So if you don't have proper planning, you will waste your time, you will waste your energy. Okay, so all of these are blended together. You have to practice everything together, not just one thing and then the rest you leave it. So that's why it's a combination of all these traits, right? Okay, I think I have mentioned about all of this. Systematic, meticulous, discipline, looking, creative and innovative. So this one is something to do with how you manage your work. Sometimes you have to be, you have to think extra. You cannot just think about based on whatever plan. So sometimes things not going based on your plan. So you have to be creative. Macam mana nak, to solve the problem. Okay. All right. Behaviors and ethics in research. So of course, this one is something to do with the ethics. Okay. So as an ethical researcher, we cannot plagiarize. That's one example. We cannot in the lab, for example, we have to be responsible. That's one thing. We have to also respect other users. We have to respect the seniors in the lab. Who are the seniors? The postgraduate. They are doing MSc and PhD. They are also working in the lab. So means that the lab is shared between the undergrads and the postgraduates. So sometimes you have to respect their time as well, respect their space. Okay, so you need their advice a lot in the lab. But in order to get the advice, you have to ask nicely. You have that one is, I think, common sense, right? So you have to develop the interpersonal skill, right? How to interact with seniors, how to things like that. I think that one, you know, lah. But that's part of the ethics as well in research in the lab. Because why it's very important? Because if let's say you create problems in the lab, so what happens? It will affect your work. So everyone will not like you, and then you'll get troubles in, you know, in your work. So that's why I said it's very important to also respect people in the lab. Okay, don't use other equipments, other stuff without permission, things like that lah. Common sense one. Okay, responsible in the lab. Being responsible in the lab means like, so when you assess the risk just now, for example, you know certain chemicals are hazardous. So maybe your friends do not use that chemicals. So you cannot just like let the things, you know, like maybe on the bench like that. So you have to be responsible because you know the things are hazardous. So you have to take care of what you are using so that it will not give hazard to others. Yeah, things like that. And then responsible, especially when you use consumables in the lab or maybe glassware, wash after you use, okay, clean the area after you use. That's part of the responsibility. Okay. Yeah. So that's in the lab. So when it comes to writing or communication, so the common issue is plagiarism. Okay. So you have to practice the right way of writing. So I've mentioned in second module, right? Plagiarism means that when you don't cite the original author and when you copy and paste the same sentence. So that's plagiarism. So you have to avoid such things. Okay, so that's it for today. Yeah, I have some homeworks for you. Rephrase the following sentences. So these are the two sentences from, I think I wrote these sentences. So you have to rephrase these two sentences, meaning to say that you have to change the sentence into a new types of sentence, but maintaining the same points. Okay. Yeah. So those are your homeworks. So this will help you to understand what paraphrasing is. Okay, that's it for today. Thank you for your attention. So basically we covered two modules. The first one is modern scientific investigation. What are the important steps? And in the fourth module, we talk about thesis research project. What is a thesis research project? What are the steps, the principles, and the characteristics, the behaviors, and ethics? Okay. So by right, you should know what is thesis, what thesis means, and what are the important aspects of it. Okay. All right. That's all for me today. Thank you very much for your attention. I'll share back the attendance. Are you guys in the campus?
It's not clear.
Okay.
So you have other courses that have physical lectures?
Yep.
Yes.
Yes.
Oh. Okay, so if you can leave the meeting. I'll see you guys inshallah next week.
Okay.
Thanks,
Thank
Doctor.
You're welcome.
Thank you.
Welcome. Welcome. Have a nice day. Yeah.
I would like to ask regarding about the poster.
Yes.
For my group, our result in the article are too long. Is it okay if we like ignore some of the results?
Yeah. Yeah. So you just decide, maybe based on your reading, right? You understand that certain things, you identify which results are the most important. So you only feature that results in your poster.
Okay, right. Thank you, Doctor.